Hi, I'm Terry Bolt, and I'll be presenting our paper, Pooled Order Rectangular Testing for Improved Public Health Screenings. This is joint work with Yuanyang Zhuang at the University of Colorado Springs. Is presented as part of the 9th IEEE Conference on Healthcare Informatics. This work was supported in part by the National Science Foundation, who was nice enough to let us take our human subjects work for another project that we couldn't complete during COVID and switch over to do something on COVID here. I also want to thank our students, Adi Nashkar, Keith Paulson, Nick Windesheim, and Stephen Joe for their contributions and discussions in the coding and some of this in testing, and then Crystal Vander Zander, who was a biology faculty who helped with some of the early work on this project. So we got to do testing. We're doing lots of it. And what is pool testing? Well, pool testing was introduced by Dorfman in the 1940s to improve testing efficiency by pooling materials from multiple samples. And basically, it tests the pool of each positive thing only if the pool if the pool that they show up in is positive. So to give you an idea, we take that rack of samples, we start collecting them, and here we're going to do this. I'm just going to show you one, but there's 24 pools here. So we take the first test tube, we put it into the pooled sample, second material from the second, put it in the test tube, third, fourth, and fifth. And so now we have a pooled test with 5 things. And now you can do this for all 24. If we do this and we go do the testing out of those 24, we might, in this case, get 5 positive pools. And now for each of those 5 positive pools, we go back to the underlying samples, we test them, each of those individuals, so there's 25 of those, and out of those, in this case, 5 positives show up. So with 120 samples with a positivity rate of 4.2%, the optimal pool size turns out to be 5, and we get 24 pools, and then we end up testing on average, there will be 5 pools that are positive, so we have to have 25. We get on average 49 tests versus 120 if done individually, so much more efficient. So an important question early on in our work, which we ended up not doing, but others that already were pursuing, was can we actually do pooled testing for COVID? And the answer was yes. Yellen et al. Was one of the early papers to show that for RT-qPCR, you could actually do up to 64 samples. So they sort of have a fluorescence plot here, and you can sort of see the purple line here is COVID positive plus 64 negative samples, so pool size 64, and it's still well detectable above the noise floor, which is way down here. And in the range of the variations you might see, here's a COVID standard template, so a little bit higher threshold, but in terms of the number of cycles, but not too much. The FDA ended up approving Dorfman-style pool testing under emergency use authorization so we could get more efficient testing. Next question is, can we actually be more efficient? So an idea that's also been around for many years is the idea of double pool testing. And double pool testing basically breaks things up into 2-dimensional grid, which you can sort of see on the matrix on the right, where we both have rows and column pool tests. And an individual sample now contributes to both a column and a row, and you test all the rows and columns. In this case, you can sort of see red for various rows and columns that tested. And then again, where, in this case now, when both the row and the column test positive, you're going to go test those individuals. So in this case, we still end up with 25 individual tests, but 22 simple tests, so we only get 47 as a total. A little bit more efficient. Double pool testing is much more efficient when you get to very small numbers, and various groups researchers have looked at what's the optimal pool size. And for Dorfman style, it turned out to be this function based on the log of 1 minus p, the positive probability, and where w is a Lambert W function, which has a sort of exponential decay. Double pool testing is more complicated, but it can be numerically computed. But in both cases, they have this very sharp drop-off as you leave very small positivity rates. So, and the question is, can we do better? So our insight really comes from the fact that not everyone in the population is the same. Prior work has all randomly sampled all of the subjects in the pools yielding performance that was based on average positivity rates. But different groups of people will have different positivity rates. Symptomatic people that have been in contact with known COVID carriers are more likely to test positive than symptomatic people who have had no contacts, because they might just have a cold or the flu. And they're still more likely to be than from asymptomatic people that have had contacts, who are more likely to test positive than people who are asymptomatic and have had no known contacts. And there's other ways you can also look at this in terms of it's known to vary by zip code, by the number of symptoms people have, by job functions. There's lots of ways we can break it down. But the question is, can we use the estimated subgroup probabilities to sort and separate things so we can use larger pools for the groups that have lower probability of positivity? And there's a number of ways you can do this. You can just apply this directly to Dorfman, and I'll show some results with that in a bit. But the technique we developed called PORT, pooled order rectangular testing, takes this and we build a two-dimensional grid like in double pool testing. But the first thing we do is sort of put all the low probability people first. We sort them in either rows or columns. In this case, we have the same 120 population we had before, but now that's actually broken down into two subgroups: 100 asymptomatic people that have 0.01 as their positivity rate, and 20 symptomatic people with 0.2. And this is just a simple example to work through. It's pretty straightforward. We do this, we end up with two positive rows because there's only one positive person on average out of 100 people with 0.01 positivity rate, and then a lot of people down here. And so we basically have these long rows which are good for low positivity rates and short columns which are good for higher positivity rates. And this allows us to deal effectively with a mix. So we end up in this case having only 10 individual tests that are needed because we only have two rows even though we have 5 columns. And this allows us to reduce our total cost down to 36 compared to 47 for double pool, 49 for Dorfman, and 120. Now to figure out what the optimal design is, we need some approximations. Since we only have approximate probabilities, we want a simple approximation. The paper develops this approach, which is really makes some independence assumptions and allows us to estimate the expected number of rows and columns given the various probability rates of every individual sample, and then you put that together to get your optimal design. So now let's look how well these things work. So this is a Dorfman style test, and there's a bunch of variation because here we have mixed populations with different numbers, and so there's a bunch of random variations you expect. The lower probability rates is a population that has mixed higher variance between the low and high probability systems, so you get larger variance than you do over on the right-hand side. But overall, Dorfman does pretty well. Double pool testing, standard double pool testing, does better at low probability rates, but actually does worse at higher overall positivity rates, so it's a mixed bag if you want to switch over to that. PORT, which is the core algorithm from this paper, you can sort of see there's a lot more variance, and part of this has to do with as we're not using great estimates of probability, we're just sorting stuff on these estimates, and so you get these sort of different clusters as you have different mixtures of probabilities and a lot of variance because there's a lot of noise in what we're doing. But almost every one of these red cells is above the Dorfman line, and almost all of them are also above the double pool testing. So overall, it's still much better. We can actually do the ordered testing on Dorfman style. There's these brown dots, and you can sort of see there's some of them that are doing very well. There's some that are actually worse because if you get your sorting bad in Dorfman, you end up with the wrong optimal pool size, and you get some bad results that can come out of that. So you have to be very careful, much more careful. And the same is true with order double. You can do it, but none of them are as good as PORT, so we don't see a lot of reason for that. We have released our code as an open source code. You can get the web version. You can actually don't have to run the code at all. You can just upload and download CSV files to get results. So if you're a lab that wants to start using this effectively, you can just contact us. You don't have to contact us, just use it. You can also build your own because lots of labs don't want to have external networks going in and out. You want to be careful what you do with your data. So you can basically set it up. It's designed to work with Docker. You download it, you build a Docker, you get a Flask container, somebody can just run on a local machine for you. And they basically use read and write CSV files with sample IDs, outputs. We're working on it so we can actually integrate with an app to collect some of the data, but for right now, we just wanted to get this out here. So, you could basically, the interface is simple. You upload a CSV file. CSV file has probabilities, rough probabilities, sample, if it's positive or not, eventually that'll get filled in, and then some IDs and some user keys. It then comes up with, in this case, a 2D design for you. This one is 6 columns by 20 rows. It's a little more efficient to show them in the screens that way. After it's run, you do your tests. You can come back and say, okay, what rows and columns were positive? You give it the same file you had before, and it'll come back and tell you the following individuals should be tested individually, and these are the same kind of examples we sort of showed before. So conclusions and future work. Sorting by expected probability makes a difference. As we saw, even for Dorfman, it makes a difference, but using the PORT approach was much more efficient. We've done testing with millions of random trials drawn from lots of configurations with a wide range of probabilities, and the efficiency gains and estimation formulas have been proven out. They work pretty well. Overall, PORT was used only 87% of the number of samples needed by double pool testing, 59% of that from Dorfman's single pool testing, and 37% of standard individual testing. So compared to individual testing, this is almost 3 times more efficient in terms of where you're going to go. Now, there are some future work we have to think about. First, we want to point out that incorrect probability estimates never impact test validity, only our efficiency gains. Right? By the time things get done, you're testing anybody that showed up in the right pools, and so you're going to get there. There is one concern on false negative, because if I have a false negative on a pool test, it can impact if individuals are detected, since we can then skip the row or column. So one of the things we do in the software is if only one row or one column tests positive, which is going to happen if you have very low positivity rates and very low viral loads, then we test everyone in that row and column because in the corresponding one that did go off, because we know it should have intersected something, we just don't know where. But false negatives are also functions of pool size and test sensitivity, so more work is needed to sort of develop the experimental validation for the maximum effective pool size for a given test. For now, our software allows users to set it based on their tools, and we recommend you be conservative. The RNA-based testing showed you could use pool size up to 64. Our software defaults to a limit of 32 for added safety, and it's really hard to effectively get to very large pool sizes. In terms of positivity rates, we have to be very low. For other problems where this might be used, that could be effective, but for COVID, this is where we are. So thank you for your time. Go back to our first page, and if you have any questions, feel free to email us, contact us if you have any questions. I'm tbolt@uccs.edu. Thank you.